So the parts in the male reproductive system include, let us start and see each and every part in detail. They include our scrotum, testis, epididymis, vas deferens, urinary bladder, prostate gland, seminal vesicles, anus and penis. So these are all the various parts that are present in the male reproductive system. So when we start with it, then among all the sex organs of the male reproductive system, the pair of testes becomes the primary sex organs, whereas the accessory ducts and the glands, they become what we call as the secondary sex organs, and the penis becomes the external genitalia. So what we are going to do is, we are going to study each and every organ in detail, and histology of the testis also in detail, so that it is more clearer for us as a clarity. So first we will start with the primary sex organs, and the main primary sex organ here is testis. Now testes are actually called as testicles, and these are a pair. The testes are always present as a pair in a two number, and they are smooth, oval, pinkish male primary sex organs. Now each testis, it has got a length of 4 to 5 centimeters long. It has got a diameter of 2 to 3 centimeters, 2 to 3 centimeters in diameter, and it has got a mass of 10 to 15 grams. So this is the external, the length, breadth, width and the mass of the each testis, and they are always present as a pair. Now these testes, they are actually present in an abdominal pouch, and this abdominal pouch near the abdominal cavity, in fact it is present outside the abdominal cavity, it is called as the scrotum. As you can see clearly, outside you have this scrotum, pouch like structure, inside which you will have the pair of testicles. Now this pouch or the scrotum, it is a loose muscular tissue. It is made up of, it's a muscular, it's a highly muscular in nature. It has got a loose skin outside, and this muscular structure has a scrotal cavity inside. It has got a scrotal cavity which is filled with a coelomic fluid. It is filled with coelomic fluid. Okay. Now, the scrotal sac has two main functions. The first function is to protect the testis or the testicles from any mechanical injuries. That is helped by the presence of this so-called coelomic fluid, which acts as a shock absorber. The second function, in fact this is the main function, is to maintain the low temperature necessary for spermatogenesis. In fact, for spermatogenesis, what is spermatogenesis? Spermatogenesis is nothing but formation of sperms. Let us write it here. Spermatogenesis is nothing but formation of sperms, which is nothing but formation of the male gamete. For that to happen, the temperature of the scrotal sac should be lesser than the internal body temperature. How much less? Somewhere between 2 to 2.5 degrees less than the internal cavity temperature, and that is somewhere between 34.5 to 35 degrees centigrade. So this temperature is maintained by the scrotal sac, and to maintain this temperature only, the scrotal sac is present below the or outside the abdominal cavity, a condition which is called as dissentation of the scrotal sac. Now, if you can see in this diagram, because scrotal sac is outside the abdominal cavity, there will be a canal which will be connecting the scrotal sac to the abdominal cavity. This canal-like structure here. Okay, this is the scrotal sac, and here you have the abdominal cavity, and you have this canal which is an empty path-like structure, and this canal is called as the inguinal canal. Passing through the inguinal canal, you have a thick cord or a rope-like structure which is called the spermatic cord. Now, the spermatic cord, though appearance-wise it looks like as one homogeneous structure, in fact if you look clearly in this image, there are multiple colored wires entangled around it. Now, each spermatic cord contains arteries, veins, nerves, the vas deferens and other tissues. Now, arteries carry the oxygenated blood, veins carry the deoxygenated blood, the nerves carry the the nervous impulse. The vas deferens is connected to the testis. It is coming from the testis and its main role is to help in the transport of sperms, movement of sperms. So all of these are present in the spermatic. Okay, now this spermatic cord runs all the way from the abdomen down to the testicles through the inguinal canal. So if I take my canal like this, inside my canal I will have my spermatic cord which is a combination of all these four, the arteries, the veins, the nerves and the vas deferens, all of them along with certain tissues. It runs all the way from the abdominal cavity to the testicles. Okay, so now let us see with the testis. Now, before we understand the histology of the testis, we have already seen what is their shape, what is their length, mass in the previous section. We are now going to talk about the important concept. What is holding the testis? Now, testis is present inside the scrotum. Each testicle is present inside the scrotum. So, somebody needs to position it inside the scrotum. So, there are two things that keep the testis inside the scrotal sac. One is the gubernaculum or the gubernaculum. Another one is called the spermatic cord. We have just seen the spermatic cord which holds the testis from the upper side. Now, what we are going to see next is the gubernaculum. The gubernaculum is actually a fibrous tissue. It's a fibrous connective tissue or a fibrous muscle-like structure which is present at the base of the scrotal sac. This is the scrotal sac, right? So, at the base of the scrotal sac, you will have this fibrous tissue or a cord-like structure which holds the entire testis. This is the testis, right? So, it is holding it from the downside, and that is the gubernaculum. So, together spermatocord from the top and gubernaculum from the bottom, they are putting the testicles in their position in the scrotal sac. So, let us see some MCQs based on the topics that we have discussed till now. What is the fibrous cord that connects the testis with the bottom of the scrotum? Vas deferens, epididymis, gubernaculum, seminal duct. The correct answer is gubernaculum. Now, next we are going to understand the function of testis, and before we understand that, let us study the internal structure, that is the histology of the testis. And once we are clear with the histology of testis, then we will go into the actual function of testis, that is spermatogenesis, which is nothing but the production of the male gametes. And also the second one, that is production of the testosterone hormone. So when I have a lateral section of a testis, this is how it will look like a mesh of wires present in some ball-like or oval-shaped structures. Now, let us see each and every part of it. Testiculars, they are enclosed. First of all, if we go from outside to inside, testis is enclosed by two protective layers. The outer protective layer is called as the tunica vaginalis, which is nothing but it's a mixture of epithelial tissue and connective tissue. So epithelial tissue and connective tissue together form the outer layer, which is called as the tunica vaginalis. Then you have an inner layer which is called as the tunica albuginea, which is made up of the fibrous connective tissue. So two layers, tunica vaginalis and tunica albuginea, form the first two layers of the testis. Now, in some books, they give you a third layer, which is tunica vasculosa, which is has areolar tissue. Some books give it the third layer, the innermost layer or the third layer, which is called as the tunica vascularis, which is made up of areolar connective tissue. Areolar connective tissue, and mostly this area consists of in some books it is given as vasculosa also. There are multiple names, okay. So it mostly consists of the blood vessels, and this is the innermost region. Right, so these are the outermost layers. So let us see the layers. The greener ones is the tunica vaginalis, and what is that? It is a pouch. It is made up of peritoneal layer, and it acts as a covering to the testis. It's a serous membranous layer or a peritoneal layer which acts as a outermost covering to the testis. Next, you have the 𝗘𝘀𝗃-𝗪𝑏𝑔enia. We are able to highlight the entire 𝗘𝘀𝗃-𝗪𝑏𝑔enia for you in that yellow coloration. So that is the 𝗘𝘀𝗃-ἀλγινία, which is the fibrous one. If you see this, the 𝗘𝘀𝗃-ἀλγινία is getting extended inside. It is not only staying in the outside, it is getting extended inside. Okay, so when it extends, what it is doing, it is dividing the testis into various partitions. Each partition is called as a septum. So each of these partition here is called as a septum or a lobule. They also called it as lobules. The walls, the 𝗘𝘀𝗃-ἀλγινία inside is called as a septum, and these partitions are called as the lobules. So testis contains many lobules which are formed due to the septum created by the 𝗘𝘀𝗃-ἀλγινία. Okay. Now, how many lobules are there? There are almost 250 testicular lobules in each testis, which we have highlighted here. Now, if you go back to the previous diagram, there is this pink space which we did not speak about. Actually, it is this pink space which is the tunica vasculosa, the third layer which contains the blood vessels. So, as we have discussed, there are 250 lobules in each testis, and each lobule, if you can see each lobule, it contains these coiled structures. These coiled structures are called as seminiferous tubules. In short, it is written as SFT. It will be easy for you to remember that. So, each lobule contains 1 to 3 highly coiled tubular structures called as the seminiferous tubules. So, let us see what is this. So, now we are going to talk about those structures, those tube-like structures called as seminiferous tubules. So, what I'll do is I'll take a lobule and I'll cut it transversely. So, when I cut it transversely, as we have discussed, each lobule consists of 3 seminiferous tubules. So, this is 1, 2 and 3 seminiferous tubules or SFTs in one lobule. So, this seminiferous tubule, it is lined by an outer lining which is called as the germinal epithelium. Inside inside the seminiferous tubules are also called as our SFTs. You have 3 main cells. First, you have the basement membrane, just like anything, you will have a basement membrane. Then the main cells are the Leydig cells, the spermatogonium, and the Sertoli cells. So these 4, if you do not consider the basement membrane because it is just a supporting membrane, the 3 main cells of these SFTs are Sertoli cells, spermatogonium, and the Leydig cells. But in this diagram, along with them, you can see structures like this which are called as spermatids. Then you can see another big structures, cells like which are present near the epithelium called the secondary spermatocyte, and the layer near the spermatogonium which is called as the primary spermatocyte. These are nothing but the order, let us say A, B, C. So these are the orders of the various stages formed of the spermatogonium during the spermatogenesis. And these structures here, these tailed structures, they are nothing but the sperms. And each of the SFTs, you can see it is hollow inside. There is a cavity that is present inside. So let us talk about each and every cell clearly. So the germinal epithelium, each of them is. So the first we are going to talk about is the spermatogonial mother cells. Spermatogonial mother cells are these spermatogonium, these cells which are lining the germinal epithelium. This is the germinal epithelium, right? So these ones that are lining it, let me take another color so that it's not in contrast. Okay, so this is my germinal epithelium, the blue one that I am drawing, and the green ones, the green highlighting areas that I am highlighting here, this is my spermatogonial mother cells, or also called as my spermatogonium, and these structures are actually our sperms. Okay, now what are these spermatogonial cells? Spermatogonia, it is actually the cells that produce the primary spermatocytes and that undergo meiosis, finally leading to the formation of spermatozoa or sperms. Now let us understand this process clearly. Normally, when we are in the embryo stage, when the fetus is in the embryo stage, at that time, due to the chromosomal constituency, it is already predestined to be a male fetus or a male child. Then there will be something that are forming here, which is called as the primordial germ cells. Primordial germ cells. Now, once this, what happens is during the childhood, during the development, during the embryogenesis and during the fetal development, this primordial germ cells, they will form spermatogonia. Spermatogonia. And these spermatogonia, they remain dormant or inactive during the early childhood. But they will become active. When they will become active? They will become active in the later stages after hitting puberty. After they hit puberty, there will be the process of spermatogenesis, which starts with meiosis and mitosis. So in this, what happens? First, the spermatogonia, they undergo various stages of spermatogenesis to form the primary spermatocyte. This is in order of maturity. Okay. Then they form the secondary spermatocyte. Okay. Now, this secondary spermatocyte further develops into spermatids. Let me take another color. They will develop into spermatids. And spermatids will finally develop into sperms. So this is the process of spermatogenesis. This whole thing is nothing but spermatogenesis, which starts with the spermatogonia. So the spermatogonia are the main gamete producing cells. We can call them as the main gamete producing cells. Next, we have the Sertoli cells. Sertoli cells have another name. They are called as nurse cells. Nurse cells or sustenacula cells. Sustenacula cells. Sustenacula cells. Okay. Now, normally these cells, they arise from the basement membrane. They arise all the way from the basement membrane, and their main role is to provide nutrition to the spermatozoa. What they do is they provide nourishment to the spermatids, spermatozoa, spermatocytes and all. And they also produce an hormone called inhibin. What is inhibin? Inhibin is a hormone which stops the secretion of FSH. Now, what is FSH? FSH is follicular stimulating hormone. So, what inhibin does is it stops the secretion of the FSH. Sorry about that. It's like a tongue twister. FSH. And inhibin also has another role. It regulates the effects of testosterone. Another very important male hormone, testosterone. Testosterone. So that effects of testosterone is also regulated by the Sertoli cells. Also, another function of Sertoli cells that is very rarely spoken about is it helps or it controls the movement. Controls the movement of spermatogonia. Spermatogonia and sperm cells. As we have discussed, this is the lumen. This here is the lumen, right? So the sperm cells have to be released into the lumen. So their movement in the lumen is also controlled by the Sertoli cells, and that's why they are called as the nurse cells. Now, after these two, in between there will be these spaces. See, this is seminiferous tubule 1, seminiferous tubule 2 and seminiferous tubule 3. So, in between these seminiferous tubules, adjacent to them, you will get these cells-like structures. Okay. So, these interstitial spaces, they are nothing but the regions present between these lobules. So, this entire space here, this entire white corridor here between the seminiferous tubules, this corridor is called as the interstitial space. Let me write it here. 1, 2, 3 seminiferous tubules, right? So in these interstitial spaces, you will find a specialized group of cells which are called as Leydig cells. So Leydig cells are a group of cells which are present in the interstitial space, and that's why they are also called as interstitial cells of Leydig. Their main role is the production of androgens, and the most important androgen they produce is testosterone. And testosterone has, what is the main role? It is the main role in providing the masculine development and also the sexual drive in men. So the main testosterone hormone comes from the Leydig cells. That's why I've told you that there are 3 cells which are very important: Leydig, Sertoli and Spermatogonia. Spermatogonia forms the male gametes. Sertoli cells nourishes those gametes, and Leydig cells produces the main testosterone, without which there will be no masculine development, no development of the male characters, and even there will be a problem in the sexual drive of the male. So let us once again understand this testosterone hormone, even though we have studied this in the chemical coordination chapter. So testosterone is an hormone that controls the development of the secondary male characters and spermatogenesis, and you can also add the point, it initiates the sexual drive in men. Okay, so that is the main role of testosterone. And who is producing this testosterone? Leydig cells are producing these testosterones. Okay, other than this, there will be some other cells also which are immunologically important. Now, if you see all these seminiferous tubules, 3 of them, they join together. Can you see here? All this is one lobule. So there are 3 SFTs and they are joining together here. Okay, so this region is called rete testis. Okay, so rete testis is nothing but it is present in this mediastinum, or in the middle of testis, which is nothing but a back mass of connective tissue. And in this connective tissue, what happens is all the seminiferous tubules of one lobule, they join there. They form a network. Now what happens here? If you look at the entire rete testis, it is nothing but a group of SFTs collected from each lobule. From here, from here, from here, from here, from here, from here. So rete testis, basically what it does is, it is an area where all the SFTs will join together and form a network. And from there, they open into a structure which is called as vasa efferentia. So they open into a combined structure called as vasa efferentia. So where is this vasa efferentia? If you can see, this region becomes our vasa efferentia, and this entire region is the epididymis. This is the head of the epididymis. This is the body of the epididymis. So the sperms, once they are matured, the sperms, what happens? They move from lumens of SFT to rete testis. From rete testis, they move into vasa efferentia. And from there, they move into the epididymis. Okay, so this is our vasa efferentia. Right? So this is our testis. Now, there is some important topics here that we need to understand. Now, I told you that the process where the testis with the coming down, or the movement of the scrotal sac rather, outside the abdominal cavity or below the abdominal cavity, it is called as descending of the testis. Basically, it happens in the fetal stage only. But there is a condition called as cryptorchidism. This is called as undescended testis, means the testis does not descend from the abdomen into the scrotal cavity. They will be always in the abdominal cavity. Now what happens if the testis is in the abdominal cavity? The temperature will be equal to the internal body temperature. As a result, they do not produce sperms. We have already discussed for spermatogenesis to happen, the temperature in the testis should be 2 to 2.5 degrees lesser than what it is in the internal body. That happens only if the testis descends into the scrotum. In this condition, it does not happen. So there will be no production of sperms, hence the male becomes sterile. So let us see some questions that are associated with the primary sex organs of the male reproductive system. In each testis, the number of testicular lobes are 350, 420, 250, 500. The correct answer is 500. Okay, so in the next session we are going to talk about vasa efferentia and the other which all and the vasa differentia, all the other accessory ducts and even the glands that are involved in the male reproductive system. So the next part of human male reproductive system is we are going to once again understand the rete testis and vasa efferentia. So as I've said in the previous section when we have discussed these convoluted tube-like structures, they are called as the vasa efferentia. And vasa efferentia, they are actually nothing but they are a common openings of the rete testis. Now what is rete testis? All the seminiferous tubules, they open into a network-like structure there, which we are highlighting, and this network-like structure is called as rete testis. This network-like structure further opens into the vas efferentia and which in turn opens into the epididymis. So there is your vasa efferentia and that is your rete testis. So both of them are connections of the seminiferous tubules from each and every lobule, each and every seminiferous tubule from each and every lobule is going to join to rete testis and from there it is going to become into vasa efferentia. From there it opens into a structure of efferent ducts. They move into a coiled efferent ducts in the epididymis and form a tube-like structure called epididymis. So the vasa efferentia leaves the testis to open into a narrow, tightly coiled tube. So this is something you have to understand. It's a tightly coiled tubular structure and that structure is called as epididymis, which you can see very clearly here on the highlighted portion of the diagram. So it is always located on the posterior surface. Remember this point, it is always located on the posterior surface of the testis. So here is an epididymis, and epididymis, if I have to divide it, I can divide it into three parts: head, body and tail. So the main epididymis, the main function of it is it acts as a storage space for sperms and it gives sperms time to mature. So the maturation time of sperms, how much maturation time? 14 days. In fact, sperms take a period of over 14 days for maturation. So that is period is given. Along with it, it can store sperms for almost several months if they are not ejaculated. So as I've said, they are divided into three regions. So let us say that the region that we are now showing it, that highlighted region is called as the head epididymis or the caput epididymis. The next region that we are going to highlight with the red color is the body or the corpus epididymis. The third region highlighted in the black color is the tail epididymis or the cauda epididymis. Now, from cauda epididymis, we have highlighted here a long tube-like structure. This long tube-like structure is called as the vas deferentia. Now, the head or the caput epididymis, it receives sperms from the vasa deferentia. So, this is the head, right? So, what happens? This is the head region. So, the sperms will come from vasa efferentia into the caput or the head epididymis. And this is a mass of connective tissue at the back of the encloses the radia testis. So there is something called as the mediastinum testis. What is mediastinum testis? It is a mass of connective tissue that is present at the back of the cell of the rete testis rather. Now after this, we are going to go to that long narrow tube, this long tubular structure, we call it as vas deferens here, which is emerging from the chorda or the tail epidermis. So this long tube-like structure is the vas deferens, or also called as the ductus differentia. Now what is it? It is a very long muscular tube that starts from the end of epididymis all the way it goes, all the way it enters into this region. This is called as the ampulla, and this is here the seminical vesicle, and this is the your vas deferens. Now vas, it passes through the urinary bladder. So this is the urinary bladder here. So what it is doing, it is passing through the urinary bladder and it is forming a common opening or a common ductus like structure with the seminical vesicle. So this is your vas deferens. Okay, so it starts from the chorda epidermis, as we have said, the tail region, and it is lined with pseudostratified columnar epithelium and lamina propria. What is lamina propria? It is nothing but it is an areolar connective tissue. So as we have discussed, it starts from the chorda epidermis, it passes through the inguinal canal. So here this is the inguinal canal, right? So it passes through the inguinal canal, through the abdomen, it loops over the urinary bladder. So here is my urinary bladder. So it is looping over the urinary bladder and forming a common opening with the seminical vesicle. It takes a duct. See here, this is the seminical vesicle, and here there is a duct for the seminical vesicle, which is called as the ejaculatory duct. So it takes from that, and the vas deferens and the duct of the seminal vesicle, it unites to form the ejaculatory duct or the ductus ejaculatus. Now, this seminical vesicle, the total length is 45 centimeters. In fact, it is quite long. It is 45 centimeters, and it has got two main functions. The first function is it also stores sperms. Just like epididymis, it also stores sperms. Okay, and the second one is it conveys, what it does is it creates a peristaltic movement, and during its peristaltic movement, what it does is it moves the sperm. It creates a peristallis movement that moves the sperm from the epididymis to the urethra. During sexual arousal, it creates that movement. So this is our vas differentia. So next we are going to talk about this ejaculatory duct. So what are ejaculatory ducts? Now, ejaculatory duct is actually ducts that converge in the center of the prostate and they open into the urethra. So here is our ejaculatory duct, which is shown. So what is happening here? The main function of it is it carries sperms from the vas differentia, the vas deferens or the ductus deferens. So what it is doing, the sperms are coming like this. So it carries the sperms like this. Okay, then from the seminical fluid, seminical vesicles, there will be fluid, right? So the sperms will mix with the fluid here, and then they are transported into the urethra. Understood? So the sperms are coming from the vas differentia, the fluid is coming from the seminical vesicles, and the sperms get mixed with this fluid. Along with that, the prostate gland also produces some another fluid which also gets mixed up here. And everything, basically the sperms will mix up with the different fluids of the glands and forming the seminal fluid, and then it moves through the urethra out of the body. Okay, so in fact, ejaculatory duct is not just a duct for the seminical vesicle and the vasa differentia. It is also a ductate for the prostate gland. Even the prostate duct also opens into the ejaculatory duct. It's a damn tongue twister. Now, these ejaculatory duct, they have a length of 2 centimeters. Okay, and what is formed by the time everything moves out of the ejaculatory duct, your sperms are filled with the seminal fluid, all the fluid material and together they form what is called as the semen. So what will come out now is the semen. Okay, so what we are going to now understand is we are going to see how the spermatozoa are getting passed. So first they will start in the seminiferous tubules. From there they go to the rete testis. From there they will move to the vasa efferentia. From the vasa efferentia they go to the epididymis. From there they go to the vasa deferens and they go into the ejaculatory duct. In the ejaculatory duct they become into semen. They become semen filled with the fluid. And where is the fluid coming from? Two ways the fluid is coming from. One, it is coming from the prostate gland. Another fluid is coming from the seminal vesicle. Seminal vesicle. Okay, so both of them produce the fluid where the sperms will mix with it and form the semen, and semen will come out of the urethra. You always have a pair of vasa deferens. Okay, and finally from the urethra they'll reach the vagina of the female. So let us see some MCQs now. Epididymis is located on anterior surface of the testis, posterior surface of the testis, ventral region of the testis and dorsal to the testis. The correct answer is posterior to the testis. Okay, so now we are going to go to the next topic, that is the urethra. The urethra is also called as the urogenital tract, which is highlighted in the diagram. Now, it is a shared terminal duct of both reproductive and urinary system. So, two mechanisms happen together in the urethra. That's why it's called as a urogenital tract. A, urine excretion of urine. So, removal of urine. So, it's an excretory system. It's a part of the excretory system. B, it is used for the ejection of the sperms. So, it's a part of the, it's a genital organ. So, it's a part of the reproductive system. That's why it's called as a urogenital tract. So it originates from the urinary bladder. So this is our urinary bladder here. So the urethra originates from the urinary bladder and it passes through the penis. Okay, and it opens outside the external meatus called urethral meatus. Now, what are the male external genitalia then? There are only two male external genitalia. One is the penis and other one is the scrotum. Now, we have already studied about scrotum. Now we are going to study about the penis, which is also called as the male copulatory organ. Now, as we have discussed, this penis is extra-abdominal in origin, that is, it is present outside of the abdomen, and it serves as a urinary duct and also as an intermittent organ that transfers the spermatozoa to the vagina of the female. So it has both functions basically. Function 1 is to carry out sorry for that. Function 1 is to carry out the excretion of the urine. So it serves as a urinary duct also. And second function is it carries out or it helps in the removal of the excretion or the ejection of sperms from the male so that it lands into the vagina of the female. So that's your sperm. That's your penis. Sorry, it's a cylindrical organ. It is cylindrical in shape and consists of three main regions: the body, the glans, penis, and the root. Now, if I cut a penis into transverse section, we see that it contains three columns of tissue. The upper column, which is highlighted in the black, this upper column is called corposa cavernosa, which is filled with which is paired and it is upper and it is dorsal in origin. So this is your corposa cavernosa. The next one, which is highlighted down in the blue color, is corposa spongiosum. Basically, it is single structure and it is ventral, it is lower side in the position and it covers the urethra. So this bilobed structure here is the corposa cavernosa, and this single structure here is the corposa spongiosum. And how can you see this? When I cut it transversely like that, you can observe it. Now, the corposa cavernosa is made of special tissue and it is filled with blood. So what it does when it is filled with blood, it helps in the erection of penis during the sexual intercourse. When there is a sexual arousal, the penis gets erected, so that it helps in facilitation or insemination. So for insemination purpose, the erection will happen when it is filled with, gushed with blood, and that is why corpus cavernosa has rich blood filled spaces. Okay, now next is the glans penis. Glans penis is this one, this bulb-like tip, the head of the penis, this bulb-like region here is called glans penis, which is nothing but it is an enlargement, or it is an extension of the corpus spongiosum. So corpus spongiosum enlarges and forms into a bulb-like structure or a head, which is called as the glans penis, and this head is covered by a foreskin or perpusate. Now let us go understand what are the various genital or accessory glands that are present in the male reproductive system. Now basically the male reproductive system has a total of 3 accessory glands. Okay, so let us see each one of them. The first one is called as the seminal vesicles, the second one is called as the prostate gland, and third one are called as the Cowper's glands, or also called as the bulbourethral glands. So these are the 3 main accessory glands or sex glands or genital glands that are present in the male reproductive system. So here is our seminal vesicle, here is our prostate gland, and somewhere here in this region will be our Cowper's gland or the bulbourethral gland. So let us see each and every gland in detail. First one is the seminical vesicles, as highlighted here. There are a pair of organs which are present posterior inferior to the urinary bladder. So they are present posterior and inferior. It means below to the urinary gland. They are tubular in nature. Now what does the seminical vesicles do? Seminical vesicles mostly secretes alkaline viscous fluid which performs 60% of semen. Now I told you that semen is nothing but the fluid which mixes with the sperms. Now 60% of that fluid is coming from the seminal vesicles and it is alkaline in nature. And this fluid contains the following. First, it contains fructose, protein, citric acid. Along with it, it contains inorganic phosphorus, potassium and prostaglandins. Now what are the roles? Fructose acts as an energy source for the sperms. Once the sperm moves out of the body, it requires energy source. Sperms move by taking the tail of the sperm. When it moves, it takes a lot of energy, and energy comes from food. So for production of ATP, you need food, and fructose acts as a main energy source. The second one is prostaglandins. What prostaglandin does is they cause the mucous lining of the cervix to be more receptive to the sperms. The cervix is associated with the female. So during your insemination, the cervix is made more acceptable, more receptive to the sperms, so that by peristaltic movement and contractions of the uterus, the sperm moves easily towards the cervix and reaches the ovary via the fallopian tubes, so that it fertilizes with the ovum. Okay, now the next one is the prostate gland. The prostate gland is this tiny gland-like structure which we are highlighting. It is a singular gland, and it is, we have highlighted it, and it is present just below the urinary bladder. Now the prostate gland, if you can say, literally it looks like the shape of a golf ball. Okay, it is the size of a golf ball, in fact. Okay, now this prostate gland, what it does is the rest 15 to 30% of the semen, 60% comes from the seminical vesicles, the rest of the fluid of the semen comes from the prostate gland. Now the main role of prostate gland is, what it does is it prevents, it is slightly clear and it is acidic in nature. And why is it acidic? What it does is it activates the spermatozoa. What it does is it activates the spermatozoa and provides nutrition, so that there is the fertilization. Now before we go into Cowper's gland, there is something that I want to mention here. The fluid of the seminical vesicles, they are alkaline in nature, whereas the fluid coming from the prostate gland is acidic in nature. Now, seminical vesicle fluids are alkaline because they want to neutralize. What this does is it neutralizes the acidic environment of the urine, acidic environment of the urine, and also the female vagina. So that is negated or neutralized by the alkaline nature of the seminal fluid, or the fluid that is coming from the seminal vesicles. Whereas the prostate gland fluid, that is acidic. Why is it so acidic and why is it got phosphate? The one reason that people say it is it acts as an antibiotic to destroy the bacteria. So to destroy the bacteria and protect the sperm from any other acids it is present. That's what one of the rules it says. Other than that, it is rich in citric acid. This is mostly rich in citric acid, which is used by the sperm to generate ATP for the movement of it. Okay. Now one more additional thing is prostate gland, it keeps increasing in size from puberty till you reach the 30 years. It keeps increasing in size. Now once you reach somewhere between 30 to 45, the size of the prostate gland is stable. But after 45 years, there is a chance of enlargement of prostate gland in males. Alright, so this is our prostate gland. So next let us go to the 𝗘𝐻 gland. So there is the location for the 𝗘𝐻 which has been highlighted. Now 𝗘𝐻 gland is also called as the bulbourethral gland, and it is almost at the size of peas, green peas. It's the size of a peas. Okay, it is just below the prostate gland at the beginning of the internal portion of the penis. And what it does, it also secretes alkaline fluid which lubricates the urethra and acts as a flushing agent. What it does is it removes any urinary residue that is present in the penis before the ejection of the sperm. So it flushes out the urine so that the urethra and the penis and the canal and the tracts are clear for the movement of sperms. So these are the three accessory glands and their functions. Now let us see the MCQs in the respect. First one, lubrication of penis is due to bulbourethral glands, Sertoli cells, Leydig cells, or interstitial cells. Answer is bulbourethral glands. Okay, so this is the male reproductive system in detail. Next, we are going to study about the female reproductive system.